Okay, thank you very much. Well, I'm taking over this lecture of Judy Williams, who is very, very sorry that she's not able to be here. And I really like to take the opportunity to also introduce a little bit more of the background of our Dementia Research Oxford initiative. Well, the whole background of this initiative is that every year there are 480,000 new patients with mild cognitive impairment and over 200,000 new patients with dementia in the UK. And the impact of dementia is enormous for the patients, the relatives, and society. So dementia by now is a major cause of morbidity and mortality in the elderly, and the economic costs really exceeds that of cancer or heart disease or stroke. So the state of the art is there's no effective treatments. The amyloid antibody trials are not convincingly positive, but also not convincingly negative. And there are many, many targets for therapy, and that came up during these discussions, but translation is incredibly slow. So that was the background that made us decide to assemble the Dementia Research at Oxford initiative, and that aims to accelerate translation and empower trials. So we have different targets that we are addressing. First of all, we'd like to improve the drug target treatment success rate and use the power of the Targets Discovery Institute and or PET chemistry to do so. We really want to improve the knowledge that we have of the molecular pathology to early detection, diagnosis, and molecular stratifications. And that is more or less the topic that we are addressing now, in addition to our efforts to develop more successful and powerful intervention and prevention trials. So what is our approach to bridge this translation gap? And I think we've been working on that for 2 years now, is that we think we should do so by a more dynamic way of translational research. And the classical functional research that leads to translation is just to start from a target, do the functional studies based on genetics, modified models, and take that to clinical care. And we introduced more or less the idea of this idea that we should also accelerate this by reverse translation and take our research in clinical care and population health to functional research also. What further is a pillar in our research is clearly that we want to start from multi-omics high dimensional studies, particularly UK Biobank that it was mentioned, but also the Million Women study and other large at-scale cohorts, and combine the knowledge that we get from data integration really with deep phenotype cohorts, clinically, cognitively, but also based on imaging, and translate in that way new knowledge about biomarkers, either in CSF or plasma, used genetics and proteomics and metabolomics, the power of the large studies, and translate that to the deep phenotype studies, and take this forward to the next level. So that involves a lot of people already, and we're having monthly discussions, and you're very welcome to join that also if you're from outside Oxford. Well, back to the title of this presentation, and why do we start from genetics if we're thinking about translation in dementia? Well, actually, if you think about dementias and Alzheimer's disease in particular, it's one of the most heritable diseases that we know of. The heritability in early onset is about 80%. Later onset, we still are facing estimates to 40 to 60%. So in the past decades, we have seen huge successes in unraveling the genetics, and that also has moved our knowledge about the disease enormously forward. And 3 decades ago, when I started my studies as a PhD, young PhD, on Alzheimer's disease, we really thought it as a neuronal disease in which we looked at the features, the amyloid plaque that was more thought to be neuronal of origin, as well as the tangles that occur in the neurons itself as the primary cause. Of course, that changed when we found apolipoprotein. Most of the research focused on the astrocytes, and more recently, due to the bulk of genetics that now almost collectively point to the microglia, it came to the attention to us that this relatively rare cell in the brain may be an important driver of pathology of this disease. Well, which pathways came up? I think it took a while before we started seeing the pathways that we were used to in Alzheimer's pathology, amyloid and tau, that came out also in the common forms of the disease, but now we're seeing them clearly in the last paper that we now published in Nature Genetics, that these are important pathways that we cannot discard from a genetic perspective. But the other pathways that popped up were clearly lipids, and I'll go into that more in detail today, but also the immune system, starting with the innate immune system, as in many diseases that emerged in the genetic association studies, but more recently also TGF-beta and T-cell pathology. Another pathway that came up was endocytosis, and if you look more in detail, ubiquitination. So if you think about the disease in the population and about stratifying the risk of the population, it is clear that apolipoprotein E is one of the major drivers of the risk in the population. What you see plotted here is by age, from 60 to about 100, the cumulative incidence of Alzheimer's disease stratified by APOE. And what you see clearly that those who carry the APOE 44 variant of apolipoprotein E, it's about 1% of the population. They have not only a higher risk of the disease, by age 90, 80% of the carriers have either developed Alzheimer's disease or have died. But we also have a much earlier onset. So if we look at the average age of onset, that is around 80 in the E44 carriers, while 95 is the average age of onset in the 3-3 carriers. So that is rather an important driver that is almost the elephant in the room in not only Alzheimer's research, but dementia research at large. So what we do, if we also weigh in the other genes, and at this moment we know about 80 genes that have been involved in Alzheimer's disease, you see that these other genes move the risk or determine the risk as well as the age of onset of disease. And this is a paper we published in 2018, but we're redoing this and it looks very much the same. What you still see that even if you weigh in the genetic risk score or a polygenetic risk score over the APOE gene, it is still the APOE, the 2 carriers, having the lowest risk in green here, depicted that never reach the high risks that are seen in the APOE 44 carriers. Although the other genes do matter, they will determine both your age of onset. If you carry a lot of risk alleles about 5 to 6 years earlier, even in E44 carriers, compared to those who are carrying the low risk variants predominantly. So what we also see in these graphs is that the genes predict whether you're developing disease, but not so much very precise when. So if you are an APOE4/4 carrier, you see the disease risk increasing already by age 60, 65. By age 70, you see a clear number of patients arising, but your onset may be as late as age 90. And that is what we usually think of, that genes determine your risk, but they don't pull the trigger when, definitely predict very precise when you are developing the disease. And that is what we would like to know in different settings, if we think about translational research, but also in trials, in how can we improve these biomarkers, both of the risk of the disease, but also the progression of the disease. Once you develop symptoms, how fast you progress, and also the progression after the diagnosis of the disease. What is also very important, and that has been lacking for long, but is now really possible to study at scale, is connecting these multi-omics data on genetics, but also proteomics and metabolomics with the epidemiological risk factors that are known, and they are very well characterized in the past decade, with early life risk factors such as education, midlife risk factors such as hypertension, alcohol use, obesity, perhaps also hearing loss and brain injury, and typical late life risk factors such as physical activity, although as Rory pointed out, that may be also a factor that may be rather a consequence of the disease than a cause. But also smoking still is a risk factor at late life, and diabetes. Well, it has come up again and again during this day, and rightly so. We also need to combine these multi-omics data to clinical aspects. So at MRI, at PET scan, but also cognitive function will be an important outcome if we think about translational research, and definitely also in future trials. So I'd like to go over with you an example, and that's an cross-omics approach that we followed using the metabolomics data that are available in the UK Biobank. And the reason for that is that we were very much interested in the metabolic changes, because if you consider the patient in the decade before the onset of dementia, you see that there is quite some evidence of metabolic changes, both in the metabolism as measured with metabolites in the blood. The glucose levels clearly go up in the later stage of the disease, but there's also quite dramatic changes seen, for instance, in weight and blood pressure, which tend to decrease before the onset of the disease. Well, that combination is a bit unexpected from a population-based perspective, because if you think that the weight is decreasing, you would definitely expect that also the glucose decreases and not increases, as we see in Alzheimer's patients. Now, we have been involved in a rather large number of metabolomic studies before we embarked on the UK Biobank data. And what we had found already in various studies, both conducted in Europe, in the mainland, but also in the US by Rima Kadura-Duk, is that you also see a clear change in branched-chain amino acids, and particularly valine and leucine are changed, reduced in patients who are developing dementia and Alzheimer's disease. Further, what we see is clearly fatty acids are reduced, and that is another phenomenon that we see. Now, when starting these studies, we really always thought, well, we see these metabolic changes, but we never connected the dots between them, and we never really studied the interplay between them if you think about the age and the major genes involved in the disease. So that is what we started evaluating in UK Biobank, 125 1,000 persons have been characterized when we conducted this study. There are still ongoing studies on the others. There's about 50,000 that are older than 60 years at baseline, and about 1,000 of them develop dementia. 553 Alzheimer's disease, 298 vascular dementia, and a large number unclassified. And what we used to characterize the metabolome is the Nightingale's platform, which measures 145 metabolites with NMR, which is a very quantified direct measurement. However, it is also, on the other hand, rather limited in that it really focuses on a limited class of metabolites, being mostly lipids and ketone bodies, and some measurements of amino acids. So we analyze the data using a classical approach that we would also follow in genome-wide association studies. We just did a Cox proportional hazard model. But in this case, because this is not a genetic factor, which is written in stone and never changing, at least during your lifetime for the genome, we had to adjust for potential confounders. We thought of as ethnicity, age, sex, and BMI. We know that BMI is a major driver of the Nightingale platform, but also technical aspects such as fasting time that may, of course, determine your levels of metabolites in the blood, but also assessment center and beds and spectrometer that we adjusted for in these data we got pre-access to. We also adjusted for potential confounders, and this is very fantastic that we used the UK Biobank data, because it also gives us an idea to adjust for nutritional, dietary risk factors such as alcohol intake, but also smoking, education, and medication, which is a very important determinant of the metabolites. So these metabolites are measured in blood, and I like to focus on that because I know there's a lot of hype about the blood-based biomarkers, but I think also rightly so, because what we actually now for the first time could do is using this platform and compare that to other studies of about similar size that we had collaborated in, and which were based on meta-analysis. We could see that if we use the UK Biobank and compare the findings, the direction of the association that we find to the study of Tinkinen et al. That used the same platform in mostly epidemiological data also, is that the findings are really very, very consistent across studies. And that is not only reassuring, it means that with these sample sizes, we have reached the stage that we can get reliable data. Well, we find novel findings. So although the findings were in the same direction, they're clearly novel, significant findings that are picked up. There are two clusters that are completely novel, and one is the cluster that includes the ketone bodies and citrate and glucose. And there's another cluster involving small HDLs. So what we did next is ask ourselves, well, if we study these incident patients, these patients are diagnosed in a period of 10 years after the assessment. And for clinical trials and translation, this is a very relevant period, because patients will often be included in this period in a trial. But the question we had is are these metabolites also relevant for the risk of the disease? And to that end, we started integrating this data in which we compared the effect that we saw for each of the metabolites to dementia to that what we saw on MRI in those who did not develop dementia. So we used the data of those who were not demented during follow-up and studied whether the metabolites were associated to hippocampal volume, which is an important outcome related to the risk of Alzheimer's disease, and total brain volume, which is a determinant of dementia in general. So what we basically see is that if we associate these two estimates and adjust for the fact that many of these metabolites are very much correlated with a mixed model, we do find that strong evidence of a correlation between the two, with p-values from 10 to the minus 14 for hippocampal volume to 10 to the minus 31 for total brain volume. What we also see that in the extremes are again the ketone bodies and the HCl values for hippocampal volume, and on the other side the fatty acids as potential protective factors for both. So hippocampal volume, of course, is that small that is a risk factor of Alzheimer's disease. You expect inverse. The same for a total brain value, citrate, and pops up with the ketone bodies as risk factors, and the branched chain amino acids and the HCl small HCl fractions as protective factors. We did the same for cognitive function, and again we see a positive line if we take reaction time, which is a measurement for executive function, which is very significant, with again the ketone bodies popping up as associated with increased risk and prolonged reaction time, and for fluid intelligence, which is a measurement of global cognitive function, we did not find a significant association. Now, what we have to realize if we interpret this data, that apolipoprotein E was not only a strong driver of dementia, but it's also a major determinant of most of the metabolites. So if we associate all the metabolites to APOE4, there's only 4 metabolites that are not significantly associated to APOE4. And for APOE2, we also find only 4 ones that are not FDR significant. It's specifically APOE4. If we do a polygenetic risk score for all the Alzheimer's genes, we see that it's still a strong association of the risk score to the HDL-VLDL axis, as well as to some of the ketones and the branched chain amino acids, but it's less overwhelming as with ApoE. So what we did next is started looking at the association of AIDS to these major metabolites that are changed. And here are 2 examples of valine and beta-hydroxybutyrate. And valine is a very well-established association. We see always see across all studies that valine levels in the blood are decreased in patients with dementia. Now, if we look at what happens over age, baseline age, these are cross-sectional data from 40 to 70 years in UK Biobank, we see that overall age ranges, the ApoE22 carriers have a higher valine levels than the 4,4 carriers here in blue, or the 3,3 carriers in green. However, what we also noticed, so that is in line what we expected, because we think valine levels are decreased, and as E4 are have an increased risk, E4 carriers have an increased risk of dementia, we expect them to have lower valine levels. What surprised us a little is that if we looked at the effect or the levels of valine in the ApoE4 carriers and 3,3 carriers over age, we see that after age 50, these levels start declining. There's also some evidence in the 2,2 carriers that they are leveling off, but less steep. Now, I'm very much aware that there is a lot of evidence of selection of people who entered the data set. However, it's very difficult to see that that would have affected differentially the 2-2 carriers and the 3-3 carriers and the 4-4 carriers. So this at least is an observation that I think that requires further study. Now, the beta-hydroxybutyrate levels, we find that they are increased in APOE 4-4 carriers, and that surprised us a little, because I think, as you may know, that beta-hydroxybutyrate or the ketone bodies are often thought of as a health beneficiary metabolite, and a lot of the diets that we follow, that are followed nowadays, are aiming to increase ketone bodies, and it's quite counterintuitive to interpret this as a risk, because that means that all these diets that we are following should increase the risk of dementia, and that cannot be the most logical explanation. So over the age range studied, we find E4 have higher ketone bodies, and then we started thinking, should we study this as a risk, or rather say, like Rory also referred to, see this as the part of the disease process, which is metabolomics, but also proteomics, will have the same phenomenon, that the proteome and the metabolome will adapt, trying to buffer the disease pathology that will be happening in APOE4 carriers long before the disease onset. So what our standing hypothesis is that if we think about the reason why beta-hydroxybutyrate may be increased is that it may point to the fact that the liver is compensating for a relatively energy deficiency in the brain already at early age. Now, that raised questions, and I think many of you will ask us, how do you know that this is because of the brain? Because if you think about this compensation mechanism, it may also be that there is vascular pathology or kidney pathology or problems with sarcopenia that explains why the liver is producing more ketone bodies if these diseases are associated with dementia. So that is definitely the case for the heart, and there's quite some evidence for the kidney pathology as well. And we started adjusting for that and could not make this go away. So starting to think about that, there's also more and more data emerging, and this is one of the more recent studies, that if you think about aging in APOE4 mouse models that do not necessarily develop Alzheimer's pathology, but they show that in the brain there is really a bioenergy deficit. Now, the fact that we found citrate and the fact that we found the ketone started pointing us to that the mechanism may be that we're looking here at a mechanism that is very much related to both acetylcholine A and the TCA cycle. And that fits with the idea that the fatty acids are low. So the fatty acids are entering the ketogenesis or the TCA cycle. You expect them to become lower in the blood. And you expect also in that case that the VHDL-HDL triglyceride axis is affected. What we also see compatible with the TCA cycle, so energy availability mechanism, is that valine is low, while glucose is high and citrate is high. So these three axes really point to the fact that there is a problem with the energy metabolism that is reflected in the blood of the patients. And that raises really the question, because if you think about the high glucose, if we really think of diabetes as a disease, that is usually associated with high valine levels in the blood rather than low. So this is a mechanism that you start thinking of as independent. So if we think about the hypothesis, we think that there is what we pick up with this platform is an energy metabolism problem in the blood that reflects the need of energy in the brain. And if we look in the brain, and we did this with the brain MPD data, then we see in the brain that valine is high, suggesting that valine is transported from the blood to the brain. But ketone bodies are not consistently, but in general, there is evidence that these are low. And that raises the question, is there a problem in the transport due to the blood-brain barrier or other reasons that the ketone bodies are not transported to the brain? Of course, there's also ketone bodies generated in the brain itself, and that is the task that is of the astrocytes in the body. So that gave us the idea that we should look at the functional studies, and we did this in a study using the iPSC cells that we have available to us that are part of a consortium of the Innovative Medicine Initiative called ADAPTED. And the aim of that consortium was to understand the function of APOE, and by studying in iPSC, two different lines. So there was one healthy donor who was a 3-4, and one AD patient who was a 4-4. And what was basically aimed to do is construct isogenic. So the genome is similar except for APOE. These patients were either not turned into a 4-4 for the 3-4 donor, or a 3-3 and a 2-2. And for the 4-4 donor, the cells were also transported to the 3-4, 3-3 and the 2-2 samples. Further, there was a knockout, and the knockout was generated to study the function of APOE, and that is what I used in this presentation. Well, what is very special about this study, we constructed different cells. So that was macrophages, the microglia, the astrocytes, and the neurons for the two donors. So what I show here for the microglia to the left and the astrocytes on the right is the evidence that there are, if you knock out APOE, there is evidence for either genes that are changing in mitochondrial function in the coenzyme A or TCA-related metabolites. And what you see there is, if you look at the genes that only show at least one-fold increase and are FDR significant, there are two genes popping up in the microglia, CHCHD2, which is a Parkinson-involved gene, and SLC16A6. But in the astrocytes, there are multiple genes that are solid carriers and transporting either glucose or ketone bodies or both into the brain. And predominantly they are lower expressed if you are knocking out the APOE, if you introduce APOE. So is this relevant? Well, I think bioenergetics is one of the drugs targets that has multiple trials on their way, both in phase 1, 2, and already 3 in phase 3. So I think one of the ideas that we can pick up, building up on Martin Landry's presentation, is that we can use this measurement or improve this measurement to be used in trials to evaluate whether these bioenergetics interventions improve better in those who are at low energy metabolism. So in sum, what we can show that there is an energy imbalance based in the patient in the decade before the onset of the disease. We really find evidence that age and APOE4 is involved in the energy metabolism, and this may be relevant for early interventions, at least to improve the patient's health status, but also the success perhaps of trials. But above all, this shows that multi-omics study have the power to identify genetic and metabolic markers in the blood for future translational research and maybe trials. So this is a start with the metabolomics data, and of course we cannot wait until the proteomics data become available for further analysis, and that can further improve our knowledge of the different pathways that are relevant for the future. With that, I have to say thank you for the Nightingale UK Biobank Consortium that was led by myself and Rima Kadura-Duk. And most of the work has been done in Oxford by June New and heavyweight lifter and Najaf Amin. And the Adapted Consortium that is really continuing despite the lack of funding, but we still are very much on the go analyzing data. Thank you for your attention and the funders.
Thank you, Cornelia. We're running a little bit over time, but I let that go because I think you got lots of interesting new data. And what I might do is therefore just try and compress some of the questions into one or two, and we could ask you about them. So I think some of the questions really relate to the relationship between APOE genotype and the metabolic changes you found. Do you think those are causal? Do you think that some of the variance is explained independently by the metabolic factors compared to APOE? Is there a mediation analysis between APOE and the ketone body levels, for example?
What we have done is both a Mendelian randomization, and there is no evidence strongly that the genes that explain the metabolite also associated with the disease. So we don't think it's in that sense a novel risk factor. However, what we do see that is that if we look at the mediation analysis, then if you add up all the metabolites that we identified as associated and select the ones who are that are independent, we see that there is a mediation effect. So what we see more or less that there is evidence that the metabolite may be in between the step between the disease variant and the development of dementia.
Right, so not an independent risk factor, but probably in your view anyway, mechanistically linked to the APOE genotype.
Yeah, and the phenotypes that we see around the disease onset.
Okay, and one other interesting question. So this is John Davis's one. How do these ketone levels compare to those reached during caloric restriction? We're not talking about that kind of level.
Yeah, it has been really a breaking point for us also. Actually, John, these ketone bodies, there's not a lot of measurements around that. I've been also, I was trained as a nutritionist once, but I think if you look at the evidence, if you would ask anyone what is a normal ketone level, we don't get very straightforward answers. What was also totally unknown was where the ketone bodies go up with age. And I thought that was quite unbelievable. If you have such a general use of a diet scheme without knowing how these outcomes relate to age at all, let alone APOE4. So I think that is quite into the dark.
So I think in the interest of time, Cornelia, I'm just going to say if people have other follow-up questions for you, they could write that in the chat.